In the endonuclease, restriction enzyme occurs, an enzyme from bacteria that can recognize specific base sequence in DNA and cut the DNA at the site. Bacteria uses restriction enzymes to defend against bacterial viruses. DNA extraction is a routine procedure used to isolate DNA from the nucleus of cells. When an isopropyl alcohol is added to a solution of DNA, the DNA precipitates out of the solution.
In the process of transcription, DNA is used as a template to create a single strand of messenger RNA. The four nitrogenous bases are guanine, cytosine, adenine, and thymine. In transcription, when the single strand is turning into mRNA, the letters pair up with their opposites. G goes with C, A goes with U instead of T, and for every 3 letters in the mRNA strand, those form a codon. Those codons, you can find the amino acids that pair up with the letters. From those codons, you can also find the tRNA. In DNA replication, helicase is able to unwind DNA by the use of energy equivalent ATP. DNA polymerase is responsible for forming new copies of DNA. Primase is an enzyme that synthesizes short RNA sequences called primers. Primer provides a starting point for DNA synthesis. Ligase fills in the gaps in DNA that are created when DNA is synthesized. The leading strand is the bottom strand that goes from 3 to 5, and the lagging strand is the top strand that goes from 5 to 3.
Electrophoresis is a term that describes a test that separates proteins in the blood based on their electrical charge and size. It's used to identify and measure the presence of abnormal proteins and the absence of abnormal proteins. Anophoresis is the electrophoresis of negative charged particles, whereas catophoresis is the electrophoresis of positive charged ions.
A polymerase chain reaction takes a small piece of DNA and enlarges it so that it can be studied easily.